Do you like ice cream? I do. It's yummy and nice. But where were we? Immune reactions. So, what immune methods do we immune methods do we have in microbiology to study infectious disease? Actually, we have a lot, and some of them you should already know. I mean, some agglutination. For sure, hemagglutination you saw when you studied blood group compatibility, identification of blood according to AB0 system. Precipitation, maybe you know, we have immunofluorescence, we have enzyme immunoassay, we have radio immunoassay, we have gel immunodiffusion, immunochromatography, and so forth and so on. Those methods are different in technique, they're different in the demonstration of the result, and you're very often confused with them. When you see that list of methods of tests you need to study for exam, you're saying, so really, that list we need to know that it's too much and they're so different, sir, please. But actually I will say, no guys, no, they're the same. Just try to understand it will be easier for you to study. All methods are the same. And I know you all like that phrase that central dogma of biology is DNA to RNA protein synthesis. And it's really so. And by analog with this, I will say that central dogma of immunology, applied immunology, is interaction of corresponding antibody matching antigen in the presence of complement. Just remember that and let it be our golden rule. Immunology golden rule. And it is important to have matching antigen and corresponding antibody. If you remember definitions of both from our past lectures, you will remember that antigen is a substance, any substance, able to induce immune response. And that immune response should be specific to that particular antigen. An antibody is an immunoglobulin or protein produced by our immunity in the response to the contact with that particular antigen. An antibody is specific to that particular antigen. It can bind only to that antigen. And both compounds should match to form a complex. Otherwise, complex will not be formed. If your antibody antigen are not matching, they will not bind. Even complement will not help. So the question is, why do we have ice cream here? It's just kind of attempt to attract your attention and to stimulate your memory. I like content addressable memory, and it is an attempt to make that, and I hope it will help. So just look here, what is it? We have initially two independent words, ice and cream. Here you have ice, here you have cream. They have different self-meaning. But if you will add one small hyphen, only that small thing here between two words, it will change the whole meaning. You have that tasty thing. And that connective bridge is obligate to have that idea, to have that concept. If you will take a cherry and put it on the cone, it will not fix, it just will fall down. And that ice cream is a connecting mediator, that small bridge. And of course, cherry should match a cone, because if you will take some baseball, of course it will not fix here, it will just smash the ice cream. And I think something like that you will remember when we will study some immune tests. We have wafer cone, which is a white shape, is like an antibody. Antigen is that cherry, and you need that fixation layer. It is complement. And of course, without ice cream, you cannot have that concept of complex. So interaction of antigen, antibody in the presence of complement is all you need to remember about any immunoassay. The rest you can logically think out according to name of the test, and you will understand the principle of the test, how to perform it. So how to apply that idea in laboratory diagnosis of infectious disease, interaction of antigen antibody in the presence of complement. Let's think. Do you remember that, our golden rule, yeah? So you should have matching antigen and corresponding antibody. They will form a complex if they're matching. If no, no complex, okay? So how do you understand? What is diagnosis? How can you describe diagnosis? I understand it as the some process of identification of some unknown. Something unknown should be identified. And in our case, it can be antibody or antigen. And let's pretend you already got your medical diploma. You are working as a doctor already. And some patient is admitted to your hospital with a known infectious disease. You need to put diagnosis and treat. So you examine the subject. By symptoms, you suspect that some problem in lungs is present. You think that it is common pneumonia. And you were a responsible student. You studied microbiology very good in institute. Panicker book, lecture, classes, everything was fine. And thus, you will know that the most common causative agent of community-acquired pneumonia is streptococci pneumonia. Next time we will study, but I will tell you now. Streptococcus pneumonia is the most often reason of community-acquired pneumonia. And you think that it is here, but you need to confirm, of course. You need to perform something. So what should you do? Correct, you should take samples. You took sputum, you suggest that streptococcus is here, but you're not sure. It can be some other bacteria. So in that case, you have, antigen. And of course, that antigen is unknown. You suspect that it is streptococcus, but you need to confirm. What else do you need to perform that diagnosis? Of course, you need antibody. And if your antibody is matching antigen, you think that it is streptococci, you took antibody against streptococci, they match, they form a complex. You was right. Your test is correct. Positive result you will have. But I didn't show complement here, if you can see. And it's not an error, because it's just so that test is so easy and obvious for everyone that no one will show that complement is here. It's obligate compound, and everyone knows that it is here. So in common schemes and common diagrams of any test, it will not be shown. You will have just antibody antigen complex, and that's all. But everyone knows that it is here. It's not obligate to designate it. But please remember that complement is something here between that antibody antigen complex. So if your antibody match an antigen, you have positive results, they will form a complex. If in your sample you have any other bacteria, some Klebsiella pneumonia, it's another one. Your antibody will not match because it is formed against streptococci. And of course, that compound will not fix, and you will have negative result. No complex, negative. Yes, complex, positive. That's the common idea of any serological test. I didn't explain what serological test. So, serological tests is a group of in vitro, means laboratory tests, based on interaction of antigens and antibodies. And routinely they use to identify unknown antigen or unknown antibody with the help of known antigen or known antibody. I mean, I think you understand. So you always have two key participants of the test, antigen and antibody. And one of them should be known. If your unknown compound will bind to known one, they matching. You know what compound you used, the same you have in your sample. They are corresponding. So if you took antibody against streptococci and your bacteria fixed here, it is streptococcus. If it's not fixed, it's not streptococcus. That's all. And actually, that is the predominant penalty of any serological test. I mean that if your test is negative, you will not be able to identify what exactly bacteria is that, because you will know only that it is for sure not streptococci. That's all. And it is a problem, because you cannot take any sample and say, just find what bacteria is it. No. That's the point to study microbiology, one of the points to study microbiology. You should analyze clinical findings of your subject, history, predict what particular pathogen can cause that particular disease in your particular patient. And you will make a note to your lab assistant, to your lab doctor, please check for streptococcus. And if it is streptococci, doctor will say, yes, it is. If no, test is negative. You need to send again, and of course it's not possible to list all possible pathogens. You need to understand what it can be, and that's how we're going to use serological tests. So you need to say look for hepatitis B, and yes, no, no. So immune tests are usually used to reveal a contact between infectious agent and microorganisms. Means they can be targeted to identify antibodies as well. So why serological? You're using antibodies, and antibodies are proteins found in blood serum. Serum, serological, that's an idea. So why do we have a large group of tests? Why so many? Again, it's pretty simple. It is the result of the progress. On the start, at the very beginning, we had very few tests, really few. But with the appearance of new opportunities, with the progress of technologies, with new data accumulated, initial group of tests were modified. Tests were modified, those methods. And new approaches appeared. And with the time, we accumulate a lot. Just that's all. And initially, for every method, we had, when it's appeared, we had some kind of era of rise when people tried to use that test everywhere. But very fast we had a subsequent fading, and some tests are not used at all in nowadays. But some are still in use, but area of their interest is pretty limited for every of the tests. And usually that use is due to is designated by benefits of the tests. So to understand again the key idea of immune tests, we will start with the most simple. Again, we said you already started agglutination. So regular agglutination is described as a clumping of a cell or inert particle by a specific antibody. The key moment here is that particle. Where is it? Here. You should have a particle. Agglutination should be correctly described as a clumping of particulate antigen by an antibody. And as you remember, one antibody can bind two antigens. It have two antigen binding sites. So at the very first moment, you will have one antibody and two particles clumped together. But why particle here is very important. Why do we say in particle? Because, how do you think? We have two key points. First of all, it is the size. Particle is much bigger than a common molecule. It's just easier to see. And you can see the result of the test even with the naked eye. So the second moment, at the very beginning of the lecture, we marked that actually one particle is not equal to one antigen, because more often a particle made of a number of identical molecules. And of course, every molecule will act as a separate antigen. And then that way, our immunity will recognize one particle, not as one antigen. It is a set of stacked together identical antigens. And of course, every antigen can be binded by a separate antibody. As a result, you will have a large number of different particles clumped together by antibodies. And again, the test is easily seen with the naked eye because of particle is big. And as far as we have antibody directly fixed to an antigen, the modification of that test is known as a direct agglutination. And we have two key usages of that test: bacterial agglutination and hemagglutination. Agglutination of microorganisms and agglutination of RBCs. Again, RBCs are mainly used in identification of AB0 system, and A and B are names of antigens which are present on the surface of red blood cells. That's why AB0. You can have A antigen, B, or nothing, 0. So technique is pretty simple. You need to take sample, regularly it contains antigen, unknown antigen. Put it in a drop of known serum, means contain known antibodies. And if you have something like that, or you have something like that, you have clumping. Solution is transparent and you have clumps like sand particles. Test is positive. If it is homogeneous, equal in density, equal in color, test is negative. It's simple. Again, hemagglutination is used to identify blood groups. Bacterial agglutination is used to identify serotypes. If you suspect, for example, E. coli, serotype O157H7, you need to take several serum, one against O157 and one against H7, and add your sample in every mix. If you see that sediment is formed, test is positive. You identified O antigen, you identified H antigen, you have that serotype. And as you can guess, if you have direct agglutination, we should have indirect agglutination as well. So what is indirect agglutination? It is a modification of direct, as we said. And it is the test when one component of the test is fixed to a particle artificially. It can be done by absorption or covalently, it's not important. But the particle here is not acting as an antigen. It is only a carrier. It's used as an indicator of the result. Why do we need indirect test? It is used when antigen itself is not a particle. And we have antibody, which is not a particle, and antigen, which is not a particle. Agglutination is not possible, a result will not be seen. But you need to see. Even if that complex is too small, you still need to see with your naked eyes. Thus, we can absorb required antigen on the surface of red blood cell, for example, and when red blood cell is covered with required molecule, you will add antibodies. Antibodies will clump those molecules. They will not bind to red blood cell. They will bind only to molecule which is fixed on the surface. And RBCs will clump forcedly. Those we have indirect name, indirect hemagglutination. Visually, result will be the same as a common hemagglutination, absolutely identical. But here cells are fixed in forced position, thus indirect again. Very often it is named as passive, because again the forced position. So what are disadvantages of that modification, and why that test is routinely not used now? First of all, RBCs are pretty sensitive to storage condition, and they have very short duration of a lifespan. So you need to refresh your supplies regularly. You need to have specific storage condition. And second reason, RBCs themselves are covered with antigens. They have self-antigens. And of course, some of them can interact with antibodies and influence the result. And the last penalty is ATIC compound. Like we have a lot of problems because RBCs are actually not growing on the trees. You cannot find them around. They're not growing from the soil. It is an animal cell. And to get RBCs, of course, you need to take animal's blood. And animals are alive and clever, and a lot of people are protesting against use of animals because it is some serological test. It's not actually so important. You need to use a live, clever being. So we had a lot of attempts to reduce that negative characters of hemaggglutinations, and scientists were looking for some other carrier particle, not RBCs. And actually, it was identified. Initially, we had Staphylococcus aureus. It is a bacteria which carries a specific unique protein on its surface. That protein is named as protein A. And unique property of that protein, it can non-specifically bind antibodies with Fc region. Fc region of immunoglobulin can fix to that protein A. And instead of binding to macrophages, it will bind to Staphylococcus. It's just a unique option of that bacteria. So Staphylococcus is spherical in shape. It can bind multiple antibodies. And in that manner you can have antibody as a particle. Not antigen as a particulate, antibody as a particulate. So you can add, for example, soluble toxins. They will fix multiple bacterial cells, staphylococcal cells, and you have again that set of clumps. You will see agglutination, the same as bacterial agglutination you see the slide before. And that test is known as co-agglutination if you use bacteria. But the whole idea, the whole concept, when antibody is a particle and antigen is soluble, it's kind of reversed. And antibody in that modification and similar modification is fixed to a cell. the whole idea of agglutination is turned upside down, and very often that modification is known as reversed agglutination. Means antigen is soluble, antibody is a particle. So the most intricate modification of agglutination is reversed passive hemagglutination. I will give you a second, just try to get to the bottom of the problem. Just try to explain to yourself, try to understand what it means. Reversed passive hemagglutination. I said that it's pretty simple. So let's put on shelves. First, what is important here? You can see that it is heme. Agglutination means you will use blood cells, RBCs, and of course we will use passive. Passive means that RBCs will not clump themselves. Something should be artificially fixed to the surface of RBCs. And of course, if it is reversed, then that something should be antibody. You have antibodies fixed to RBCs, because in normal direct you should have antibody as a soluble compound, but here you have antibody as a particle, thus reversed. And of course, that is the idea of the test. You need to add antigen and you will have formation of complexes tested positive. So more often that modification is used to diagnose viral disease, because viruses are too small. Even clumped together they will not be seen. And virus is a kind of a particle but extremely small. So that modification is used. RBCs are acting as a carriers of antibodies, and that is the result. So nowadays, of course, both methods are almost not used. And we have another thing. We have latex. Latex particles are synthesized artificially. They are uniformed in size. They are naturally contain no antigens. They are absolutely inert in immunology. And of course, they are much more stable. It's much easier to store them. It's just some artificial, like a rubber or plastic thing that have very long time period of storage, and moreover, it is possible to use latex as a carrier for either antigen as well as for antibody. And of course, you can provide that latex particle with some visible color. And more often, we use red and blue. Reaction is pretty easy to see. Positive test, you have those sand particles like clumps, and negative homogeneous solution. So of course, nowadays we have a lot of where are they? Commercial tests. So precipitation, it's pretty simple. Precipitation is formation of insoluble complex of initial soluble antibody and soluble antigen. No particle here. That is the key difference. Both compounds are from the very beginning soluble. They are naturally soluble. But when they will form a complex, that complex will become insoluble. It will become visible. So here we have modification which is known as a ring test or ring precipitation. To perform the test, you need narrow glass tube, and you need to fill in with the lowest part. You need to fill in with serum containing required antibodies and soluble antigen. You need to overlay both solutions very slowly and carefully. They should not mix like in a cocktail. So if you will have precipitation, if antibody antigens are matching, you will have that ring sediment between two liquid layers. And if you will take a look on the light, you will have that translucent, where is it here? You say that ring, it is positive test. It is formed. Antibodies which can perform precipitation are known as precipitants. They are able to precipitate out of a solution upon antigen of binding with antigens. And in routine practice, the test is used mainly to identify anthrax. It is known as some Ascoli test, to find out antigens of Bacillus anthracis in human tissues. So pretty easy and good seen. Another modification of precipitation test is flocculation test. It can be done on slide, it can be done in tube, so the same technique. Antigen is mixed with the drop of serum, mixed well. If you have formation of flakes like here, test is positive. If it is some equally homogeneous, test as negative. And we have the DRL test to diagnose venereal syphilis as one of the most popular and important examples of that test. So tube flocculation mainly performed in some manufacturers to titrate antitoxic serums to make toxoids or some other preparations. In common lab practice and routine diagnosis, it's not used because it is too difficult and too huge. So the most requested modification of precipitation test is immunodiffusion. And immunodiffusion is when compounds or components of reactions are added to the gel. An antigen-antibody combination or complex is formed by means of diffusion into the gel. So more often we will use agarose gel, the same as you used in electrophoresis. Gel electrophoresis, I hope you remember the same principle, the same idea. So result of that immunodiffusion will be visible like a band of precipitation. The same opaque opalescence kind of semi-transparent thing will be formed. Do you all remember that common gel electrophoresis idea, right? So again, just few and very primitive things. You need to take a gel. Initially it is solid. Melt, it will become liquid. You can fill it in any volume, container, plastic or some other, and when it will cool down, it became solid. And here in that picture I tried to show that the key idea of the gel, you have a lot of, like here, a lot of pores, a lot of cells, like channels. And the whole gel is made of those channels. And those channels are used to pass antibody, antigen, those we say diffusion. So of course it's obvious that bigger and heavier molecules will start very fast. They will move slow. And lighter molecules and smaller molecules, they will go faster and run far. It's obvious idea, but how to apply it in immunology? We have several modifications of immunodiffusion. The first one, the most simple, is single diffusion. When only one compound, antigen or antibody, will diffuse and another should be already incorporated into the gel. How to do it? What do I mean? You cook in the gel, you took that container. When you will melt it and slightly cool down, you will have still liquid broth or liquid agarose. And for example, you will add antigen here. It will dissolve in agarose. And when you will fill the container with that gel, you already will have a gel containing antigens. Antigens are already here. They don't need to diffuse anywhere. They're fixed. And when you will add antibodies in the gel, antibodies will diffuse. And if your antibody and antigen are matching, they will form a complex. You will see the zone of precipitation like here. If antigen antibody is not matching, you have nothing. Pretty obvious idea, single or simple immunodiffusion. So another modification of the test, when standard gel and you will incorporate both antigen and antibody in the gel simultaneously. Gel initially is empty. You can do it in Petri dish, for example, and when you will add antibody and antigen, they will diffuse towards each other. And again, if they matching in some zone, you will have precipitation. If they're not matching, you will have, of course, nothing. So a few examples of ready result, you see that V-like strip, positive. Otherwise else, no zones, negative. The same you can perform in the slide, for example. In one, you added antibody into other antigens. If you have some weak positive, it's poorly seen, or good positive. So another modification, third modification, is radial immunodiffusion. It is simple or single diffusion, but in 2 dimensions. I mean that antibody can be incorporated in the gel. It can be done in slide or here in the Petri dish. And antigen is applied into cutted wells. You need to remove some part of the gel, add antigen here. And when antibody will find antigen and form a complex, you will have ring around that well, the ring of precipitation. And that is again the photo of ready result. And if you can see, diameter of those zones is different. It is directly proportional to the concentration of your compounds. More antigen, bigger zone, far distance. That particular method is known as Mancini tests. And pretty simple again. So before we will move to next topic, we should mention one phenomenon. And I pretty hope again you all know what is cytolysis. We have bacteriolysis, but now we need to talk about hemolysis. For sure, again, you know that problem with incompatibility of different blood groups. And our RBCs again have A or B receptor. If you will add blood containing antibodies alpha or beta, they are hemolytic. They can destroy RBCs. And in the diagram I showed you different degrees of immune lysis, of blood lysis. Again, it's kind of obvious. So again, we're saying antibody and hemolytic, antibody and antigen, but what is not mentioned here? We said that it is immune reaction. So antigen and antibody to interact needs complement. Again, I said it's routinely not mentioned, but complement is needed. And if you have complement, your hemolytic antibody will bind to RBCs and they will be destroyed. And again, complement is obligate. Cytolysis I showed you in previous and last lecture. It is due to that C5 portion of complement. Complement will attract C5, C6, 7, 8, 9, and they will form that perforin system and cytolysis. So again, why we don't mention complement routinely? Because you're using antibodies, and antibodies are present in the serum. So when you will add serum, complement as a serum protein system. It is already here. We don't need to add it specifically in any test because it is always here in that serum you're using. So why did we say about that? We have complement fixation test, CFT. And complement fixation test is an immune test which is used to determine whether or not antibodies to a particular antigen are present in patient's serum. To evaluate the result, again, you should have limited amount of complement. Very, very few. And as we said before, in any serum and patient serum, you will have a lot of complement present. So before you will start complement fixation test, you should inactivate started serum. For that purpose, serum was heated in such a way complement proteins will be inactivated, but antibodies will remain. Again, it just was identified, it's just known that complement proteins are much more sensitive to heating. They are susceptible to temperature. Antibodies are more stable. And under certain conditions, you can inactivate proteins of complement systems, but they will not, idea will not inactivate antibodies. So after inactivations of patient serum, we can perform CFT. CFT performed in 2 steps. Step number 1, you need to take antigen, antibody, patient serum, and complement. Antigens are routinely taken as a form of diagnosticum. It is a preparation containing antigens we are interested in. You need to take diagnosticum contain antigen, small amount of complement, and heat and activated serum from patient. And now you have 2 options. Your subject can be positive, it's either positive or negative. What it means? So if you're saying that subject is positive, we mean that antibodies are present in patient serum. Again, we said that complement is used to identify antibodies in patient serum. If your subject is negative, in serum you have nothing. So let's think, what will happen if your subject is positive? You have antibodies. If you will add diagnosticum matching antigens, of course, they should bind to antibody. But again, they cannot bind directly, they cannot bind themselves. They need complement. And if your subject is positive, complement will be used to bind antigens, and we say that it should present small amount. All complement will be fixed on the complex of antigen antibody. That's why it is critical to have small amount, because absolutely all complements should be fixed. No free complement should remain. Of course, it should be in small amount and you have antigens, antibodies. If your subject is negative, what will happen? You have antigens, you have complement, but no antibodies. Of course, complements will not, that complex will not be formed because it's not possible. And all your complement proteins are showed as that some symbol. It remains free. Looking easy. Free complement is present, negative test. No free complement, positive test. Easy, right? How to see is it fixed or not? How can you see if complement is binded to complex or not? Of course, you will not see because complex is invisible. And actually, why did we talk about hemolysis last slide? Complement is protein which is soluble in solution. It's absolutely soluble, it's invisible. So to understand, to see free unbinded complement, we need some indicator. And as you can guess, if we talk about hemolysis, indicator is hemolytic system. And to see the result, we need to take red blood cells and corresponding hemolytic antibodies. And then the second step of the test, we need to incubate first portion of the reaction and hemolytic system, 37 degrees Celsius again. And if your RBCs will be destroyed by antibodies, it means that complement was free. That our golden rules, that if complement will not be fixed in the first step, if it is here, you will have antigen antibody complex formed, and you will have, of course, hemolysis RBCs destroyed. If your complement was fixed in the first step here, you will have nothing to bind antibody and RBCs, and hemolysis will not occur. You will have just soluble good blood. Of course, that result is visible with the naked eye. Hemolysis is easily identified. Even here on the photo, you can see obvious differences. So CFT complement fixation test was used for diagnosis of venereal syphilis. It is the most frequent example. But as we said already, some tests are not used, and CFT nowadays is almost not in use. Rarely we can use them nowadays. So besides obvious reasons we mentioned before with RBCs, assessment of that result is kind of pretty subjective. I mean, it's not difficult to see that difference when you have that test tube and that test tube. It's easy. But what if you have weak result, as I showed in the last diagram? Very, very, very weak positive. And you have not a single test, but you have 100 tests, or maybe 200 of them, and light is not very good, and you're working for the whole day. So I want to say that if you have a lot of tests are not strongly positive, it is a chance that some error, and the chance is pretty high that some error will be done by lab assistant, because lab assistant is very tired. And if you will miss the result, if you will not catch the result, of course, it's not good. And actually, to perform the test, you need to you need to use high amount of reagents. A lot of RBCs are needed. And nowadays, the common trend of lab diagnosis is to use micro tests, but with improved accuracy of the results. And nowadays, we have very good example, ELISA test. Some say ELISA, but manufacturer originator said that it is ELISA. And again, it's obvious from the name, it is immuno. So you already should understand the principle, because immuno, it is sorbent. So some compound of the test should be fixed on the solid phase. And you have enzyme linked means some of compounds should be linked with an enzyme. Again, if you remember that idea, it's not difficult to guess what is the idea of the test. So ELISA nowadays have multiple modifications and multiple usages. But to understand the basic idea of the classic ELISA, we will take the most simple modification as an example. It is direct ELISA, or it is immunochemistry sometimes. No, it's some like that, just to show you it's easier to see. But maybe you saw something like that in the histology department or some path anatomy class. And immunochemistry or simple ELISA is used to identify particular compound in tissue slices. And you know that in tissue slices, of course, in tissues we have multiple different antigens, proteins, hormones, some receptors, enzymes, and they all fixed are here. They all are fixed here. You have some set of different. And for example, we need to find human MACE1 in skin of the human. You need to take native slice, and without staining, you will have only that gray scale. It's transparent. But you know that the tissue is rich with multiple antigens. Maybe some cytokines or something else are present here, but you need to find one particular unique antigen that is already present in the tissue. So if you know that you have an antigen which is somewhere here, human MACE, we said human MACE1 you need to find. You need to take antibody against that particular antigen, antibody against MACE1 cells. And of course, if you say that it is enzyme link, you should have enzyme fixed to antibody, and you have some substrate. More often, horseradish peroxidase is used, or alkaline phosphatase, HRP or AP, and corresponding substrate. So you already have antigens applied in the tissue. You will add antibodies, and they will find corresponding antigens. They will bind and form a complex. After that, you need to wash a slice, because if antibody is not fixed, it should be removed. And of course, when you will add substrate will find antibodies with linked enzymes, and enzyme will chemically convert a substrate, and substrate will change in color. And in corresponding places, you can have some red color or blue color. You will see that your required tissue, your required some sample is present here in that particular area, and you will see approximately how much. Pretty simple technique, right? But when the technique is not very good, and why it is not good in diagnosis of infectious disease, because in clinics, more often our samples are liquid, blood, pus, sputum, CSF, urine. And if you will wash your antibodies, of course, you will wash everything. And how to stain dissolved compounds. So nowadays, more often we're using plastic. You all saw that 96 well plastic plate, it is in use. That plastic have non-specific ability to bind any biological molecule in non-specific manner. Any you will add, it will bind here. So kind of simple demonstration, you have that surface and some surface with some area. If you will add your sample, that plastic surface will absorb any antigen, and that stage is known as antigen fixation stage. So again, you need to wash unbounded substances, some non-specific debris, and in second step you need to add antibodies against corresponding antigen, again labeled with an enzyme. And if required antigen is present, antibody will bind. Again, you need to wash the sample, and if antibody is not binded, it will be washed away. But if complex is formed, as it's seen here, it will remain. And when you will add substrate in the third stage in that plastic well, solution in the plastic well will change the color, because if enzyme is present, substrate will chemically react and color will be changed. And that diagram, again, it is easier to see. Transparent, you have nothing, transparent wells, and if your result is positive, you can see that color is changed. Multiple colors we can use, blue and some red, yellow, orange, a lot of. So, of course, you should understand that if antigen is not found here, if it's absent, your antibody will not fix, substrate will not react with the enzyme because it will be washed away and you will have no color. That is again kind of classic usage of ELISA nowadays. But we said that we have multiple modifications. So if we will try to show schematically that idea, you have surface, antigen, antibody. We say that it is direct capture and direct detection. It means that our antigen-studded antigen directly captured on plastic surface and detected directly by that labeled antibody. That modification have two major disadvantages. First, you will have a lot of unneeded molecules fixed to a plastic because we say every antigen will stuck. And of course, you can have cross-reactivity because some antigens can absorb antibody non-specifically. You will have false positive result. Second problem, if studied antigen is present in very few amount, your color will be changed very few again. It will be slightly changed. And it's not difficult to miss the result, and you will have false negative result. You can see incorrect changing of color or miss the result. So to improve the quality of the assay, it was suggested to perform indirect capture. It means that manufacturer will initially cover the whole plastic surface with known antibody against your targeted antigen. No empty space will be left here, and when you will add your sample, only targeted required antigen will bind to antibody, nothing more. It will improve specificity in direct capture. So if you will take schematically, you see it resembles sandwich. You have two similar layers and one unique in the center. It's resemble that food. So that modification is frequently known and named as sandwich ELISA. If it is so, you should understand that antibody, and targeted antigen between. So to improve quality of the detection, it was suggested to use indirect detection. As you can guess, initially we have primary antibody, which is not labeled. It is used to bind to antigen. And secondary antibody against antibody or anti-antibody. It's not against antigen, it's against primary antibody. The main idea that amount of secondary antibodies will be much higher because primary antibody, again, is not one. It have multiple locus to bind antibodies, and color will be more pronounced, increased sensitivity. So, of course, we can use indirect capture, direct detection, direct capture, indirect. We can combine multiple variations according to weak side of a particular test system. You need to improve accuracy or sensitivity. So we have two modifications of ELISA, quantitative, qualitative. If all you need just to have plus or minus, you have that modification. If you need to understand how many, you can use quantitative. You will see different scales of initial color. Again, according to intensivity of the color, we can suggest how many is present in the test, how many antigens or how many antibodies are present in studied volume, and you can recalculate. Of course, you can take a look with your eyes, but again, it's difficult and your eyes will be tired very frequently, very fast. So nowadays, we have automated test systems. We have that analyzers. You can see that it has some computer. You need to put a well, and it is a spectrophotometer. You need to put on the plastic plate here, load samples, some info, and of course the device will give you much more accurate results and much faster. And chance of error, of course, is pretty low. So, of course, as we said, we can have antibodies against antibodies. So we can detect not only antigens in the samples. If we will pre-absorb some, for example, animal proteins against human IgG or IgM, You can identify immunoglobulins, and you can identify even different classes of immunoglobulins. And as far as you can count how many are present in the serum, you can identify even some approximately stage of the disease or quality of immune response. And nowadays, use of ELISA is very diverse. It's very popular method, really. And nowadays, again, we have a lot of different manufacturers, a lot of different commercial kits which are ready to use. You have a plate included here, multiple reagents, list of steps how to perform the test. You can identify IgM, IgG, antigens, and of course it is very popular method to study viral infections, bacterial pathogens. You can evaluate quality and speed of immune response. And of course, to have so many tests, you need a lot of antibodies. And some will say, aha, you're using animals again, it's not a good test. But no, ELISA uses monoclonal technology. All antibodies are synthesized artificially in the test tube. Do you remember that idea of hybridoma? And in that key, test is really good. The only thing which cannot be reached by ELISA is still preciseness in quantitative mode, because change of color is still not very perfect way to measure amount. And test is enough in medical practice, but not in experimental one. It depends on quality of the plastic, quality of the lamp, quality of the detector. And if you need to know exact number of molecules, how many till the molecule, you need another modification. But again, it's pretty easy. You can see radio immunoassay. The idea is the same as ELISA, but one significant difference. Label here is not an enzyme, it is radioactive isotope. More often, some iodine-125 is used. Of course, amount of signals is measured in gamma counter, not spectrophotometer, not your eye. It's named as RIA. It is highly sensitive. But of course, we understand that the price of equipment and of course health hazard to lab assistants argue against its use in small laboratories. And nowadays that method is used only in some scientific research, in some scientific institutes, laboratories. So one very modern technique, it is immunochromatography. I'm pretty sure you all saw some strips, plastic strips with that window here, and you can see some strips with the color that appeared across. So again, if you will try to understand the idea, it's pretty simple. It is immuno. So the test using nitrocellulose membrane, which contains fixed detection antibodies against your required antigen. In known place, you know that antibodies are fixed here. So to perform the test, you need analyte solution. It contains antibodies, labeled antibodies, and some antibodies against required antigen and indicator. Of course, you need to see. So regularly colloidal gold or latex particles are used as precipitates. They will provide the color. And you will need to add a sample. Together, you need to mix sample and analyte solution, put it in that lower zone of sample window, and after you will mix, if antigen-antibody complex will be formed if your test is positive. You will have the complex in the strip, and it should migrate toward to antibodies and a solid support by a capillary flow. And when it will reach detection antibodies, they will fix. Antibody detection antibodies will fix antigen and bind here, and you know that position. And when immune complex is formed that conjugate solution, gold or latex particles, they will generate routinely red or purple or blue lines. If you have that line, your sample is positive. No line, complex not formed, negative. The main kind of problem with that test, it's pretty sensitive to factors, proteins. Proteins are sensitive. They are sensitive to temperature regimen, to humidity, to sun exposure, and improper storage condition can spoil the test. And if your antibodies are destroyed, you never will have complex here. So you always will get negative result. Your indicator system is destroyed. So to be sure the test is performed correctly, we have another zone. In the same stress strip, we have control antibodies. Control antibodies are targeted against labeled antibodies. They are not against antigen, they're against antibodies present in analyte solution. They always are here. So when your complex or even antigens are absent, only antibodies will reach that control zone. They will stop here. And the same conjugate will give you result here. So control zone always should be positive. If it's not formed, it means that something is wrong with analyte solution, and your result is for sure incorrect. So nowadays, it is very popular technique to study COVID infection, COVID-19. You see that here you have positive test, control and test. Both are positive. Test is positive. That one is negative. Control zone is bright, but that one is negative. Test is fast, 5 to 15 minutes. And one more advantage of the test, you need very few amount of sample, only one drop, few drops maybe. Of course, the device is very portable. It's like that in size. And it can be done near the bed of the patient. You don't need in some extra equipment, expensive, not any equipment is not required. And of course, anyone can do it. It's perfect when you have distant location out of the hospital, some village, or maybe some urgent state of the subject, and you don't have time to wait for lab, or your lab is closed, or it is some traffic accidents, you need to do it now, you cannot transport your subject. And of course, even subject him or herself can perform the test. It can be used for home diagnosis. It's perfect. And of course, at the very before COVID, immunochromatography more often was used as a pregnancy test, strip to identify pregnancy. Again, you can have several strips according to some test systems can identify stage of the pregnancy, some time period. But we said that we can identify antibodies as well, and we can differentiate IgG, IgM. So modern modification of that COVID-19 system, you can have control, IgG, IgM. In that test, both are present, G and M. You can have only G, only M, or nothing. So you can evaluate immune response and approximately stage of the disease even with the help of the test. The most charming test is, I think, the most charming for me immune test. It is immunofluorescence. It's pretty long known, and it's not the latest test for sure, but it is still in use. It's good because from physics you should remember that fluorescence is a phenomenon in which a particular chemical compound named fluorochrome emits the light of a particular one single spectrum of the color after being treated with another spectrum. And as the result, you can have that beautiful, unnatural glowing. And I'm pretty sure that similar photo, and one of the most known photos is Legionella. It has the property of natural fluorescence, and it can self-fluoresce. But in medical microbiology, that option is pretty rare. So that photo, I think, is the most popular. If you will enter fluorescence microbiology, you will have a lot of different views, examples of that: night sky blue. Another example is Pseudomonas. Pseudomonas aeruginosa producing fluorescent yellow-green pigments. In next time you will study that bacteria, you will have all enzymes produced, all pigments produced, you will know. But actually, that's all only few examples of natural fluorescence we have in medical microbiology. So, as you understand, routinely we use an unnatural fluorescence. It means that some antibodies against pathogens will be labeled with fluorochrome artificially. And when you will treat your sample with the serum contain antibodies, as that in that picture, they will cover bacteria and they will repeat the shape of the bacteria. So in microscope, you will have black or dark background. You need UV microscope. It is type of dark field microscope, and microorganism glowing with corresponding color. The test is very good when you need to identify some rare pathogen. which is not possible to cultivate in routine lab, or it's very slowly growing Francisella. In that view, it is Francisella causative agent of tularemia. It's even prohibited to cultivate that bacteria in routine labs. You need to have specific permission, specific license. So cultivation is not possible. PCR is not available because it's a rare disease, but it's fast progressing, and you don't have a time to wait until a sample will be sent to some specific lab, and commercial test systems are not available. Again, plague the same problems you will have. So immunofluorescence, common serum, treat your sample. If you have that glowing color, you have positive results. So the key idea of the test, you have microbial cell, you will apply antibodies. If they will stick, you have shape of bacteria, and fluorescent labels will emit the light. Again, test is fast, 5 to 10 minutes is required, and easy to perform. You need only microscope, but routinely UV microscopes are available. And then nowadays, the test is spread far beyond microbiology. Not only bacteria can be studied with the help of the test. It is used in study of tumors. It is used in study of cell tissues, chromosomal disorders. It can be studied in. It can be used in the study of some chromosomal aberrations, meiosis, and some other processes in the cells. And in real life, it's even more beautiful than here. So it's looking really bright. It's glowing like a natural in the black background. So maybe you will like that method, and you will find yourself in lab diagnosis because of immunofluorescence. So in very fast idea, that's all we need to talk about today. Actually, even now my time is overused, and I really hope you will understand key concept of immune tests and remember our golden rule. So see you in the next lecture. Goodbye, and save your knowledges.